Good afternoon, or good morning, or good evening, wherever you are. This is the epidemiologist seminar in our weekly series, and it's a great pleasure to have you. And it's of course also a great pleasure to welcome our two speakers of today. Elkes Price is a professor of epidemiology in the Department of Epidemiology, as well as in the Department of Biostatistics. And he is in the program in genetic epidemiology and statistical genetics. You will hear what he focuses on in his research. And second speaker of today is Ben Strober, who is a postdoc in the group of Dr. Price, and they will jointly give this seminar. It's fine mapping causal tissues and genes at disease-associated loci. Dr. Price, I think you would start. Go ahead.
All right, we're going to share screen here.
Yeah.
And let me just minimize this. Let's see, do I click on this one to minimize it? How do I get that to go away? Very good. Good afternoon, everybody. So I will be co-presenting this talk with Ben Strober, who's a postdoc in our group. And here's an outline of what we'll be talking about. And I'd like to start out by saying that I think it's extremely boring to talk about published work. And with that in mind, I'm going to start with sort of a general introduction to functional architectures of complex disease. And then I'm going to delve into what is actually at the advertised title of this talk, Fine Mapping Causal Tissues and Genes at Disease-Associated Loci. That work was recently published in Strober et al. 2025 Nature Genetics. And then more excitingly, I'm going to hand the reins over to Ben, who will talk about some unpublished work in preparation on estimating disease heritability mediated by molecular phenotypes. So let's start with an introduction to functional architectures of complex disease. So genome-wide association studies have identified many tens of thousands of genetic variants associated to complex diseases and traits. And these days, if we perform a GWAS in a large sample size, in the old days, we call these Manhattan plots, but now the genome is just totally carpeted with, many loci that are very clearly and robustly genome-wide significant. And that's good, but the problem is that we generally have no idea what the biology underlying these genome-wide significant disease loci is. And so that's what the focus now is, trying to understand what all these GWAS associations are actually doing biologically. And with that in mind, I'm going to define functional architectures as sort of attempts to characterize how are the genetic variants that impact disease, how are they distributed across different functional variant annotations, for example, coding variants, conserved variants, regulatory variants, and so on. And maybe there's annotations that may implicate particular disease-critical tissues or disease-critical cell types or cell type contexts, as well as we're interested to identify specific genes whose expression impacts disease, which is a little bit different from identifying associated variants. So those are all things we try to do in sort of elucidating functional architectures of disease. And so a little background of some work that's already been done in this space. We know from work that goes all the way back to Finucane et al. 2015 that all the different traits we study are certainly enriched in coding regions. So coding regions may only sort of explain about 10% of disease heritability, but it's certainly true that coding SNPs are enriched for disease heritability. SNPs that are conserved across mammals are enriched for disease heritability. It may be varying across different diseases how much they're enriched, but they're certainly enriched. And enhancer marks such as H3K27AC are also enriched for disease heritability. Again, that can vary across diseases, but all diseases are robustly enriched. So we know a little bit about which types of functional variants variants carry more disease heritability. We also know, and this is a little surprising and maybe counterintuitive, that SNPs that have a lower levels of linkage disequilibrium, that is to say, SNPs that are correlated with fewer other SNPs, actually have larger causal variant effect sizes. And that's true for all the SNPs that diseases and traits that we've studied. So from agent menarche to unibrow, we know that SNPs that are less correlated to other SNPs actually have larger causal effect sizes. And that actually has an evolutionary interpretation, that those SNPs that are less correlated to other SNPs tend to be more recent, and as a consequence, sort of on average subject to stronger negative selection, implying that they're likely to be more functional and more disease and complex trait associated. We can delve into sort of specific disease critical tissues by looking at chromatin marks in those tissues. And that can tell us that if we're studying schizophrenia, it's SNPs that have regulatory activity in CNS, that central nervous system, or brain, that tend to be most important. If we're studying height, it's connective or bone tissue that tends to be most important, and so on and so forth. And we can also draw similar inferences by looking at a different type of data, which is gene expression data, and identifying genes that are specifically expressed in particular tissues and SNPs located in or near those genes or connected to those genes. And once again, we see that for schizophrenia, it's CNS or brain tissue that's most important. For height, it's connective tissue that's most important, and so on and so forth. And specifically, as we sort of try to zero in on the particular genes that are associated to disease, there's a paradigm called a transcriptome-wide association study. That's a mouthful, but it's basically where are trying to identify genes whose predicted expression, as predicted by the genotypes of nearby cis SNPs, whose predicted expression is associated to disease. And the TWAS framework provides a framework for doing that. And we can take that one step further, and we want to say that we want to distinguish causal genes from tagging genes whose genetically driven expression happens to be correlated to the causal gene. And this is an example here from a paper of Mancuso et al. 2019, who, and it's widely known that SORT1 is the causal gene at this locus for LDL. Both SORT1 and a bunch of other genes, including CELSR2, are strongly associated, implicated by the transcriptome-wide association study framework. But then when we layer TWAS fine mapping on top of that, we can see that the SORT1 gene attains a high posterior probability, or PIP, and the other gene, CELSR2, attains a low posterior probability, indicating that SORT1 is the causal gene at this locus. So with all this great work that's been done to sort of identify functional variants, causal tissues, cell types, causal genes, you might wonder if there's any more work that needs to be done here. And the answer, it turns out, is yes, there is more work that needs to be done. And this brings us to the second part of the talk, which is titled Fine Mapping Causal Tissues and Genes at Disease-Associated Loci. So as I've just mentioned, gene level fine mapping in the transcriptome-wide association study framework is able to distinguish causal genes from nearby tagging genes. And we might wonder what challenges might still remain in that endeavor. And so one challenge is that in addition to identifying the causal gene, we might be interested to identify the causal tissue. It could be that for a particular trait, we could have one locus where it's gene 1 specifically acting in adipose tissue that's biologically important, and a different locus where it's gene 2 specifically acting in brain tissue that's important. And we want to identify the causal gene tissue pairs or gene cell type pairs, and not just the causal genes. A second challenge is that understanding that the tissues and cell types and cell type context that we've assayed are certainly only a subset of those that are truly biologically acting. We may have a non-mediated variant. And when we say non-mediated variant, we're not necessarily saying that that variant doesn't impact gene expression, but what we're saying is that variant doesn't impact gene expression in any of the particular tissues, or more generally cell type, tissue cell types and cell type contexts that we've assayed. And for that reason, with respect to the data that we actually have in hand, we would characterize that variant as being not mediated by gene expression in the assay assay tissues. And it would be important to account for that possibility when we do our gene level fine mapping. And the third challenge at this stage in time, the GTEx consortium is widely agreed to sort of have the best, human body wide gene expression resource, but the sample sizes here are not infinite. And even for some of the more widely sampled tissues, limited sample size can sort of limit our ability to develop perfectly accurate gene expression prediction models, or CIS prediction models. And we may wish to account for the limitations of finite sample size in order to avoid false positive associations. So those are three different important challenges underlying this endeavor of identifying causal genes or causal gene tissue pairs at a particular disease locus. And there's been substantial recent work on these challenges. And here I'm going to describe the TGFM method of Strober et al. 2025, which addresses actually all of these three challenges. So this is sort of an outline for this TGFM part of the seminar. So starting with the TGFM method, TGFM stands for tissue gene fine mapping. And the idea here is, well, first of all, we're just going to use the existing transcriptome-wide association study framework to associate the cis-predicted gene expression of a given gene in a given tissue to disease by building cis-predicted expression models for each gene tissue pair. And then the sort of important bit, or the new bit, is we're going to kind of distinguish which of those gene tissue pairs that are associated to disease are sort of causal and which are merely tagging, while also accounting for non-mediated genetic variants whose association disease does not act through impacting gene expression in any of the tissues that we've assayed. And this is a model that kind of describes what we're doing mathematically, where Y here is the phenotype. The next term, G beta, is G is genotypes. That's sort of genetic contributions that don't have to do with assayed tissues. Then this next term does have to do with gene expression and assayed genes in tissues, where G is a sum across genes and T is a sum across tissues. And we have here the genotypes multiplied the sort of genetic effects on gene expression. So delta is the sort of genotype expression effects, and then alpha is the sort of expression disease effects, the impact of the causal impact of expression in a particular gene and tissue on disease. And then finally, of course, epsilon is environmental noise. And we really want to know, what are these causal non-mediated SNP effects beta, and what are these causal effects on disease of expression in a particular gene and tissue alpha? That's what we really want to know. We put a particular prior distribution on our betas and alphas, and the goal is to infer posterior probabilities, or PIPs, that quantify the probability that each non-mediated variant or gene tissue pair actually has a non-zero causal effect on the disease that we're studying. And importantly, we are allowing for multiple different causal genetic elements here. Genetic elements refers to either non-mediated variants or to gene tissue pairs whose expression is causal for disease. And one of the key aspects of TGFM, I mentioned that we're kind of identifying causal gene tissue pairs and not just causal genes, and we're leveraging data across the genome to put in priors that are tissue specific. So we can look across the genome to say that if we're studying schizophrenia, it's probably brain tissue that whose expression has the highest prior probability of being associated to schizophrenia, and we can leverage genome-wide data to identify that that's the case. And then I also mentioned the challenges of limited sample size of the gene expression resources, such as GTEx, that we're going to use. And TGFM addresses that challenge using a sampling approach, where we actually sample 100 different gene expression cis prediction models of how we can predict expression using SNPs. And because there's uncertainty in those models based on finite gene expression sample size, we're actually going to sample from from a distribution as to what the coefficients that allow us to predict gene expression using cis-SNPs actually are, and we're going to carry those 100 samples through the entire algorithm. So I'm going to gloss over some of the details here, but in terms of input and output, we input GWAS summary statistics. We do not require individual level GWAS data. We input LD, preferably in sample variant LD from the underlying GWAS, which will improve the precision of fine mapping. And then we input EQTL data from a gene expression resource such as GTEx. And then the output are these posterior probabilities of causality, both for expression gene tissue pairs, as well as non-mediated variants whose impact on the disease is not mediated by expression in the particular tissues that we have in hand. And we use the term genetic elements to refer to either gene tissue pairs or non-mediated variants. We can also compute gene level PIPs by just summing together the gene tissue PIPs or posterior probabilities across all the tissues. So we conducted a few simulations to see how well this works in a simulated setting. And we can see that TGFM, which is our method, in contrast to some of the other methods that we simulated, is able to have a sort of well-controlled false discovery rate relative to a particular posterior probability. So that's good. And more broadly, instead of trying to fine map gene tissue pairs, we can do gene level fine mapping where we sum across tissues. And we can also do variant level fine mapping of non-mediated genetic variants. And those seem to be well calibrated as well. And we can also assess power. And the power here is modest. Just fine mapping is a statistically hard problem. We see in sort of SNP level fine mapping studies that the power to fine map SNPs is generally pretty modest, and that's what we see here, that the power to fine map causal gene tissue pairs or causal genes or causal non-mediated variants tends to be pretty modest at the sample sizes that we simulated. So in general, the picture from simulations look good. Of course, we're going to want to go to real data to get a sense of how successful this approach can be. So we applied it to 45 diseases and complex traits from the UK Biobank, and we did identify a substantial number of causal gene tissue pairs, about 16 on average for each trait that we studied. So traits are listed here at the bottom. And this is for different values of posterior probability. The black lines are a posterior probability of 0.5. So 16 gene tissue pairs on average across traits at a posterior probability greater than 0.5. And then these shadings represent how well we do it at either more stringent or more lax posterior probability thresholds. Not surprisingly, if we attack the easier problem of just fine mapping genes by summing posterior probabilities across tissues, we're able to fine map a larger number of genes at a posterior probability greater than 0.5, with obviously the drawback being that then we don't know the causal tissue if we do it that way. And we also fine map a substantial number of non-mediated variants that are not, whose effect on the disease is not mediated by expression in any of the assay tissues. And we do identify more non-mediated variants than gene tissue pairs or even genes, consistent with the widely acknowledged fact that the tissues that we've actually assayed in GTEx or in other available data sets, probably span only a small fraction of disease heritability, and is probably most likely unassayed tissues, cell types, and context that sort of carries the remainder of disease heritability. So here's a few examples. We looked at the disease eczema, and sort of consistent with known biology, this OVAL1 gene in lymphocytes attains a very high posterior probability of being a causal gene tissue pair. And we also identify a causal non-mediated variant at the locus. And I mentioned earlier that we impose priors based on genome-wide data to see which tissues should be assigned the highest prior probability. And in terms of the priors that the method actually assigned, as a sanity check. We do see that for LDL, liver is the primary tissue with whole blood as a secondary tissue, as we expect. For all autoimmune disease, it's spleen and lymphocytes, which are both known to be immune-related tissues. For vitamin D levels, it's primarily skin, but also liver and whole blood. And as a sort of validation of the genes or gene tissue pairs that we've fine mapped, this slide is more about validating the genes. We used the POPs method developed by Hilary Finucane's lab, in which they use actually, they sort of predict the importance of a gene for a particular disease by training a model that uses various features and uses sort of genetic data from other loci, but not the locus of the target gene. So this is sort of an independent validation, and we were pleased to see that the genes most strongly implicated by TGFM with high posterior probabilities indeed are the genes that are most strongly enriched for having high POP scores. Finally, we applied the method to some single cell data from Presidol 2022 Science. So now we have 9 sort of fine-grained cell types in PBMC. And for example, for the trait monocyte count, we fine mapped the CD52 gene in classical monocytes, consistent with known biology. And here we also fine mapped another non-mediated SNP at the locus. More broadly, we fine mapped about 30 gene PBMC cell type pairs at a pip greater than 0.5. So conclusions of the TGFM part of the talk, that TGFM fine maps not just causal genes, but causal gene tissue pairs at disease loci. It avoids false positives due to non-mediated variants, and it also avoids false positives due to limited eQTL sample size by undertaking a sampling approach. And then in applications to real data, we identified causal genes and tissues or cell types in application to the 45 UK Biobank diseases and traits in conjunction with 38 GTEx tissues, and also in the single cell data, 9 PBMC cell types. So I'm now going to talk it over to Ben, who's going to talk about unpublished work in preparation on estimating disease heritability mediated by molecular phenotypes.
All right, so the next section is on estimating disease heritability mediated by molecular phenotypes. And to kind of transition from the previous section, one thing we can do with TGFM is ask the question of what fraction of the fine mapped genetic elements, so either non-mediated variants or gene tissue pairs, mediate a given disease. And on average, across diseases and traits, we found that only around 11% of TGFM fine map disease signal is mediated by gene expression, where the other 89% is coming from these non-mediated genetic elements. And this is consistent with other work, notably the MESC paper published by Doug Yao in Nature Genetics in 2020, which does a similar goal, which is to estimate the proportion of disease heritability mediated through bulk adult tissue gene expression levels. And they found a consistent number in that only 10% of disease or trait heritability is mediated through these GTEx bulk predicted gene expression levels. And the goal of this second section is to basically ask the question, what about the remaining 90%? 10% seems awfully low. And where is this remaining 90% of disease heritability acting through? Is it acting through other tissues and cell types or so on? And to look at this, I'm going to propose 3 possible explanations for this remaining 90%. The first is something Alkis mentioned a fair amount, which is that we actually need gene expression levels in the correct disease context. And perhaps having gene expression measured from only GTEx adult healthy individual tissues is insufficient. And we need to measure gene expression in the correct cell types or contexts that are most relevant to disease. And related to this, there's been a lot of recently published single-cell eQTL data that spans many diverse cell types and contexts. And one limitations of this single-cell eQTL data is it's of relatively low sample size, which I'll touch on in a moment. The second explanation is we need molecular phenotypes other than total gene expression levels. There are, in fact, many molecular phenotypes other than total gene expression levels that can potentially mediate disease, such as histone modifications, RNA splicing, proteomics, and so on. And importantly, for all these we have listed here, we also have QTL data for all these other molecular phenotypes other than gene expression, and we can potentially assess how relevant these other molecular phenotypes are for mediating disease heritability. And the third explanation is this more statistical explanation in that our current estimates from MESC are actually attenuated or downward biased due to finite sample size EQTL data. And the reason being is that MESC relies on genetically predicted gene expression levels. And this predicted gene expression provides noisy estimates of the true unobserved genetic gene expression levels. And because we have these noisy estimates, this will result in attenuated mediation estimates. And then they showed this analysis in simulations where they do observe a reasonable amount of attenuation in the estimate, but the extent of the downward bias in real data is unknown. And so what I want to do here is we have these three possible explanations: gene expression in the correct disease context, other molecular phenotypes, and this statistical attenuation. And I want to evaluate each of these three possible explanations for this missing regulation. And to do this, I'm going to do a twofold approach. And in the first step, I'll develop a new method to estimate the amount of disease heritability mediated by each molecular phenotype. And to do this, we'll model EQTL uncertainty and hopefully produce unbiased estimates regardless of the EQTL sample size, with the understanding that it is okay if the standard errors on these estimates get really large at small QTL sample sizes. And secondly, I want to apply this new method to jointly estimate the heritability mediated uniquely by all these different classes of QTLs, such as bulk tissue QTLs from GTEx, single-cell QTL data, as well as QTLs from other molecular phenotypes. And what this will allow us to do is basically make the following pie chart for each disease that we have GWAS data for. And the total pie in this pie chart represents the total genetic component of a given disease. And basically what this will allow us to do is basically divvy up how much of that genetic component of disease is mediated through each of these molecular QTL data types we have access to, and how much is non-mediated. And with this information, we'll be able to answer a really interesting question, like the motivation question, which is how much of the genetic component of each disease is mediated versus not mediated. And secondly, we're also going to be able to go further and ask questions like, what is the contribution of each molecular phenotype to each disease? And what is the contribution from fine-grained cell types or other molecular phenotypes? Okay, an outline for the remaining part of this section is I'm going to first discuss an empirical assessment of the impact of EQTL sample size on MESC estimates, basically assessing the amount of attenuation in these MESC estimates. And to do this empirical assessment, I did what we call, or what we're calling a GTEx downsampling analysis. And what this involves is basically starting with the full set of GTEx data, which consists in the meta-analysis of 13,000 total RNA-seq samples, and then basically downsampling those 13,000 samples on a grid from 98% all the way down to 50%, and basically assessing how different our estimates of mediated heritability are across these downsampling, effectively allowing us to assess the impact of sample size. And at least at the really large meta-analyzed GTEx sample size, the mediated heritability estimates are only modestly affected by EQTL sample size when looking at this really, really large meta-analyzed EQTL cohorts spanning about upwards of tens of thousands of individuals. But importantly, when we look at smaller sample size EQTL data sets, the mediated heritability estimates become strongly affected by EQTL sample size, and suggesting that this attenuation effect is most strongest at these smallest QTL sample size and becomes saturated in the ballpark of thousands of individuals. And with this knowledge, I still think this motivates that we need new methods to determine the contribution of these small sample size QTL data sets, such as single cell EQTLs. And also we're going to need, new methods to be able to partition the contribution of mediated heritability across all these classes of molecular phenotypes, even if one of those molecular phenotypes comes from a small sample size QTL data set. So with that in mind, I'm going to introduce a new method we're working on developing to try to get at this question of developing an unbiased estimate of how much mediated heritability goes uniquely through each class of molecular phenotype. And the underlying generative model for this is going to be similar to the one Alkes presented in TGFM, and it follows the following generative model likelihood, where our disease or trait Y, which is a vector across individuals, is decomposed into these two genetic effects. There's these non-mediated genetic effects, and there's these mediated genetic effects from each molecular phenotype M. And specifically here, G is the genotype, K indexes SNPs, so GK is the genotype of a particular SNP. Beta K here is the non-mediated variant of disease effect size. Little m indexes molecular phenotypes. Little g indexes genes. G again is the genotype matrix. Delta MG here is the causal QTL effects for molecular phenotype M, gene G, and alpha MG is the causal gene to trait effect for molecular phenotype M in gene G. And the quantity we're interested in is the proportion of heritability that's mediated by these molecular phenotypes. And so you can represent this mediated heritability quantity as a ratio of the two genetic components. In the numerator, we have the mediated genetic component, and in the denominator, we have the sum of the non-mediated and mediated genetic component. Notably, we have this term beta K, which is a causal effect, beta K, which is the causal non-mediated variant to trait effect size. The expected value of beta K squared across SNPs is equal to the per variant non-mediated heritability. So, and the expected value of alpha squared mg, so the causal gene to trait effect size across genes, is proportional to the per gene molecular trait M mediated heritability. And delta mg here, which I want to emphasize, is this unknown causal cis QTL effect size corresponding to gene G in molecular trait M. And what the previous approach MESS does to look into this is it takes a two-step approach, where in step one, they set delta mg equal to the predicted causal cis QTL effect size. And so that I'm going to call it delta mg hat, learned from a lasso regression on finite sample size EQTL data. Then they set delta mg equal to delta mj hat, and given this, assuming the causal EQTL effects are equal to the predicted causal EQTL effects, you can then simply estimate these two parameters, beta K squared and alpha squared mg, using a summary statistic-based approximation to the above likelihood. And what we want to do here is we want to go beyond this and no longer just assume we know the true value of the causal EQTL effects, but model the uncertainty in those causal EQTL effects. And to do this, we're going to introduce an approach that we're calling joint LDSC. And joint LDSC estimates mediated heritability by modeling uncertainty in both the GWAS effects and the EQTL effects. And I'm just showing it this here where we have our, basically I'm showing our chi-squared statistics for our GWAS data, and then we also have chi-squared statistics corresponding to the EQTL data for each gene. And the goal here is to basically integrate these two sources of information together, the GWAS data together with the EQTL data. And to do this, joint LDSC follows on a popular method called LDSC, or LD score regression, which basically decomposes the GWAS chi-squared statistics into a term that has our heritability of interest, which is our beta J squared parameter. And what we can do is expand upon this, similar to MESC, by replacing beta J into these non-mediated component, which I'm calling gamma J, and these mediated component, delta J times alpha G. And we can plug this in here, but the problem is we still have this unknown parameter delta J G squared here that we don't know. And so what we do in joint LDSC is we have this term from the GWAS chi-squared statistics, but then we can also do the LDSC decomposition on the QTL chi-squared statistics for a given gene, which, as you may expect, decomposes the EQTL chi-squared statistic for a given gene into the following equation, which also has this delta JG squared term. And what we can do with this is we can basically do a joint optimization of the GWAS chi-squared statistics together with the EQTL chi-squared statistics to jointly model this term, delta JG squared, as well as jointly infer these other parameters or these other heritability parameters of interest, gamma J squared and alpha squared G. But that was a lot of details, but at the end of the day, we have this new method that we're calling joint LDSC. And joint LDSC takes as input GWAS summary statistics, multi-molecular phenotype QTL summary statistics, and a reference panel LD. And takes this input, performs this joint optimization, and outputs a mediated heritability estimate for each molecular phenotype, as well as a non-mediated heritability estimate. And this is done in a disease-specific manner. So we get a specific estimate of each of these quantities for each disease. Okay, and so what I'm going to show next is this new method, joint LDSC in simulations. And the simulation has the following overview, where we simulated data with a GWAS sample size of 100,000, an EQTL sample size ranging from 100 to 10,000, and we used real genotype data from a chunk of chromosome 1, We assumed the non-mediated heritability was 0.27, the mediated heritability was 0.03, and we included 5 tissues or 5 QTL data sets in this analysis. And these QTL data sets, as expected from biology, are correlated across the QTL data sets. But in our simulations, only one of these tissues was actually causal, and the other 4 are non-causal, but simply correlated with the causal tissue. And what I'm showing here is I'm showing showing joint LDSC in simulations in estimating mediated heritability. And again, the truth here was 0.03, which is shown by this dashed line. And what you can see is joint LDSC, which is the blue bar, gets approximately unbiased estimates of the mediated heritability in the causal tissue, regardless of the QTL sample size, with the expected increase in standard error on those estimates getting larger at these small QTL sample sizes. And we compared 2 existing approaches. The first is MESC, and then a second one, which is what I'm calling 2-step LDSC, which is similar to joint LDSC, except it doesn't model the uncertainty in the QTL effect sizes. But in general, these 2 other approaches that don't model uncertainty, MESC and 2-step LDSC, get attenuated estimates at smaller QTL sample sizes, whereas joint LDSC gets approximately unbiased estimates regardless of the QTL sample size. And further, that showed for the causal tissue, but we also have these 4 non-causal tissues that have true value of 0 mediated heritability. And as expected, joint LDSC gets an approximately unbiased estimate of 0 mediated heritability averaged across these 4 non-causal tissues, whereas these approaches that don't model the uncertainty falsely attribute mass to these non-causal tissues to basically compensate for the fact that the causal tissue was not fully specified. But in general, we're really excited about this approach, and we have this new approach that we're calling joint LDSC to estimate the proportion of disease heritability uniquely contributed by each molecular phenotype. And joint LDSC produces unbiased estimates of mediated heritability regardless of the QTL sample size. And in future, we're actively starting to apply joint LDSC to real GWAS diseases and molecular QTL data sets, allowing us to determine the molecular phenotype mediated architecture of each disease. And that wraps things up. And a bunch of people to acknowledge, including Martin Zhang, Tiffany Amaruta, Jordan Rosen, Xiling Zhang, Kang-Shin Hao, and Luke O'Connor, and obviously big thanks to Alkes on both of these works. But I think at this point we'll take any questions.
Thank you very much, Alkes and Ben. This is really very interesting, and you had a good speed in there. I'm not sure I got everything, but I thought it was highly interesting. Perhaps if you unshare, we can see whether there are questions or comments from the audience. Anyone who would like to ask a question or make a comment, please?
I was wondering if you could explain a little bit more specifically how this could be applied to actual disease.
Are you referring to the second presentation, the second section?
Yes.
Yeah, I mean, the basic idea is that you have a given disease that you have GWAS for. I don't know, take autoimmune disease, and you have your GWAS summary statistics for autoimmune disease, and then you have this reference set of QTL data, spanning GTEx, but other QTL data sets, and you throw it all together into this algorithm, and the algorithm will output basically what is the mediated architecture of autoimmune disease. What fraction of the total genetic predisposition for autoimmune disease goes through gene expression measurements in T cells versus how much goes through splicing in I don't know, some other cell type, and it will basically give you these big picture insights into generally how the genetic component of diseases is organized and what molecular pathways these genetic effects go through.
And can I just add to that? If we ask what do we actually know right now? I don't know, pick an autoimmune disease, let's say rheumatoid arthritis. If we ask ourselves how much of the heritability of rheumatoid arthritis can we explain with molecular traits that we've measured, we could say that, you know, it's something like 10% of the heritability of rheumatoid arthritis is explained by expression in all of the GTEx tissues, sort of a union across those tissues. And then we can offer isolated examples where, you know, maybe somebody has done single cell RNA-seq or single cell ATAC-seq in some fine-grained cell type and found some example where it's, you know, it's this particular fine-grained cell type where, you know, where we've got some single cell EQTL or whatever. But right now, we don't have the methodology to kind of fold that into our 10% because the methodology doesn't really handle small sample sizes and correlated traits. So if you want to know how much we can add to that 10% by folding in like fine-grained single cell data at low sample sizes, the answer is we don't know. You know, we got to wait for Ben's method to answer that question.
Thank you. Further questions or comments?
Hey, that was really great.
Andrew, please go ahead.
Thank you. That was really great. I have a question. So maybe it's too early for this, but there have been numerous genetic correlations been given across many disorders that have come out, you know, the Broad and Neal group there. And I'm wondering, are you going to apply your techniques broadly to try and see if there are not just genetic correlations between diseases, but if those are mapping onto the same cells and tissues across diseases so that we know that where there's actually overlap in the tissues causing disease, not just these genetic correlations?
Well, I will say, I mean, there's been some work, notably from Tiffany Amoruda in Nature, is it 2024 or
23.
2023, where she developed this new method called TCSC, which in one aspect of TCSC was it allows you to basically decompose genetic correlations or genetic covariances between a pair of diseases across a given molecular phenotype or EQTL tissue type. And you know, you saw like, you know, certain tissues popping up that made sense to explain a given genetic covariance. But similar to the MES method, you know, this approach is affected by finite sample size QTL data. And I think you could ultimately take if you were, you know, motivated, and you could take a similar approach to what we're describing here and extend it to decomposing this genetic covariance, which is maybe what you're getting at. And I think it could be really interesting to like really get at like, you know, are there these shared, you know, are the genetic effects that are shared between two diseases, you know, mediated through the same type of molecular phenotypes.
And I think it's a good point that, you know, I mean, that this was, you know, an example of this is the work we did in LD score regression and cross-trait LD score regression. But I think it's a good point that any statistical method that we can apply to characterize the variance of a disease or trait can probably also be extended to characterize the covariance of any pair of diseases or traits.
Cool, thank you.
Thank you. Other questions? We have 1 or 2 minutes still. Perhaps I misunderstood you, but one of you, I think as Elke said, we are very interested in how this proportion 10% to 90, how that is for different diseases. Can you say something general about this?
Well, so the 10% is the proportion of disease heritability that is sort of mediated through the GTEx tissues. And, you know, that the estimates for individual diseases are somewhat noisy, but, and you can look it up in the MESC paper, Yao et al. 2020 Nature Genetics, but for most diseases and complex traits, you're at some number close to approximately 10% of the disease heritability is mediated by GTEx tissues, which is a set of tissues that, you know, have been assayed in fairly large sample size. And, you know, the whole world is wondering why that number 10% is so tiny. And the sort of the prevailing view is that, well, number one, GTEx doesn't capture all of the sort of fine-grained cell types and cell type contexts, which are unfortunately currently assayed only in small sample sizes for which the MESC method is simply not going to work. Or that we need to bring in other molecular phenotypes, histone QTL, chromatin accessibility QTL, other molecular traits will help us explain more of that. And it gets, the statistics gets harder when we have smaller molecular trait sample sizes, and the statistics also gets harder when we have different molecular traits that are correlated or genetically correlated to each other. And those are some of the issues that Ben's method is addressing.